Thanks, Kay, and thank you all for hanging out to the end of the day here. I appreciate your interest. I guess I'll start with just a brief overview of the company. I just have 2 slides here to give you a sense of what Caladrius is today. We are a late-stage therapeutics development program with 3 principal development trials ongoing in the clinic. 2 of these programs are designated breakthrough in their respective jurisdictions of development. We have CLBS 12, which is being developed in Japan for critical limb ischemia. It's in a phase 2 trial under a Sakigake designation, which is the equivalent of RMAT in Japan. And it's a trial that's eligible for early conditional approval if it shows an appropriate trend toward therapeutic effect, and could actually qualify for full approval if we show statistical significance. The trial is roughly halfway enrolled, and we expect to complete enrollment by the end of this year, have top-line data in 2020, and with the accelerated review afforded by the Sakigake designation, CLBS 12 could be a commercial product in Japan as early as 2021, and we're in deep discussions with a number of Japanese pharma companies to become a commercial partner for that product. We also have an RMAT application in for this product in the United States, and that will determine the regulatory path forward for a U.S. development program, which we hope to start at some point in the near future. Another program is CLBS 14, CMD, where CMD stands for Coronary Microvascular Dysfunction. This is a plaqueless angina disease. That is a disease where there's no discernible large vessel problems to address. So it's a disease of the microvasculature. This is in a phase 2 proof of concept study here in the United States. The last patient enrolled should be within a month or so, and we expect to have top-line data at the end of this year, 6-month follow-up. What's important, in addition to the typical clinical markers, this study also has a quantitative measurement of heart perfusion, that is coronary flow reserve. So we'll be able to correlate increase in the microvasculature within the heart with a decrease in symptoms and an improvement in exercise tolerance. And we're looking forward to reporting that study, as I said, at the end of this year or early next year. And then the study that is closest to registration in the United States is our CLBS 14 NORDA program, where NORDA is no option refractory disabling angina. These are patients who actually have both large vessel and microvasculature disease. The large vessel disease is treated, and in these patients has been treated with the traditional approaches, pharmacotherapy, interventional techniques, and cardiovascular surgery. The microvasculature component has no approved therapy, and so our Cd34 cells are what is employed here. This is a program that has an RMAT designation in the United States. It is phase 3 ready, and we expect to start a phase 3 trial in the second half of 2019. We're in the final throes of negotiation with the FDA on the design of that single trial for registration, but we've already agreed that the previously used endpoints in phase 2, such as total exercise time, will be the primary endpoints in this particular trial. The company is financially stable. We're debt-free. We just reported our 2018 results a week ago. We have over $40 million in cash on the balance sheet, burning on average about $5 million a quarter. So that will take us easily through middle of next year, and we have a long history of being able to acquire non-dilutive funding from organizations such as NIH, CIRM, that's the California Institute for Regenerative Medicine, Sanford Foundation, one of our research partners, JDRF, and others. And across our pipeline, we have a number of value-creating events which will be reported, which should drive interest and value and bring us that much closer to registration. So for CLBS 12, the RMAT results will come out probably in the second quarter. We'll finish enrollment at the end of this year and have top-line results next year. For the CMD trial, we'll complete enrollment imminently and have top-line results by the end of the year. And for the NORDA program, we'll be finalizing the plan with FDA, initiating the trial later this year. So in a nutshell, that's the company today.
Well, great. So Dave, we've heard a lot about different stem cell therapies that the different companies are working on. Can you describe your Cd34 cells and why is it those cells are best suited for the applications that you're targeting?
Certainly. So Cd34 cells are homopoietic stem cells that reside naturally in the bone marrow. Their function in nature is to promote angiogenesis of the microvasculature. So we use them for what they were intended. We harvest them in a technique, pardon me, through a technique that no longer involves bone marrow aspiration, but actually allows us to mobilize the cells to the peripheral blood by using low doses of GCSF in the donor for a few days. Those cells are harvested. They are then sent through an apheresis process. The sample of mononuclear cells are sent to a processing facility where the cells are sorted and then concentrated and resuspended for injection in a proprietary media that preserves their viability and potency, and then they go back to the center and to the patient that donated them and are injected in a site that's determined by the indication. So our CLI patients get intramuscular injections, our CMD patients get an intracoronary injection, and the NORDA patients receive an intramyocardial injection. So we're using the cells as nature intended. There's no genetic modification. It's simply collecting them, concentrating them, and putting them physically in the place of ischemic insult.
So let's talk about CLBS 14 NORDA. This is an interesting program. You acquired it from Shire. That came with a lot of clinical data, phase 1, 2, and 3. And what has that clinical data demonstrated thus far?
Well, it's interesting, and just to be clear, we acquired a data license from Shire. Originally, Shire got that through Baxalta, which came from Baxter, and originally that program was being developed at a time when intellectual property was awarded to our predecessor company, Neostem, now Caladrius, which actually effectively was blocking IP on the Baxter product. So Baxter, had they completed their program, would have actually needed to take a license from Caladrius to market that product. That data that has been generated, which has been generated in randomized, double-blinded, placebo-controlled trials here in the United States, is compelling and consistent across all the trials. I think the most interesting data is the phase 2 data, the standalone data that was from 168 patients. That data shows statistically significant improvement in exercise tolerance, which is the defined phase 3 endpoint at 6 and 12 months, so continuing to improve after a single injection of Cd34 cells. It shows statistically significant improvement in angina frequency, that is a decrease in angina frequency, a decrease in angina severity, and most importantly, going out to 2 years, a mortality benefit that is accompanied by a trend toward improved MACE as well. So really compelling evidence on all the markers that would be used in phase 3 in a population using a product, a dose, and an administration that's identical to what we intend to use in phase 3. And Baxter had started a phase 3 program, which they stopped when they decided to strategically move away from, among other things, regenerative medicine. That data has been looked at now, and that data is completely consistent with all previous data. So they would have had a positive phase 3 study, and a group at Duke, as well as a group at Columbia, have independently performed pooled patient level analyses on the phase 1, 2, and 3 data, and come to the conclusion that they are all corroborative of the benefits that I just described.
And in terms of your learnings from those studies and how that informs your phase 3 design, what are the differences that you're going to apply to your design to enhance your probability of success?
Well, there are a couple things that we'll do to, if not enhance, certainly try to ensure probability of success. And one is to make, from a perspective of endpoints and follow-up, to make a minimum of changes from the phase 2 trials, so that the phase 3 is what, a phase 3 should be, which is a confirmatory study in a large population without many new variables. So we'll be looking at the same primary endpoints that the agency agreed with Baxter as acceptable for phase 3, and those were used in phase 2, total exercise time, and a number of secondary endpoints on angina frequency and the 6-minute walk test and a few other things. We'll be, of course, using the same cell sorting technology, so manufacturing or processing will be the same. We'll be using the same administration. So this is an administration that's done intramyocardially using a mapping catheter, a NOGA system. We actually have data that suggests that intracoronary and intramyocardial injections produce the same result. But to be safe, we're not changing the method of injection for our phase 3. And we'll be focusing on that same NORDA patient population, which is a very specifically designed patient population with exactly the same inclusion criteria.
So you do have the RMAT designation for this indication. I think of interest to a lot of the other companies here. How has that actually impacted the efficiency and your accessibility to speak to the FDA about this program?
So it's an interesting question, and it's one that's difficult to answer without a frame of reference. So I can't tell you what our interactions with the agency would have been like without the RMAT designation, so it's hard to make a comparison. I can say this, that in some respects, the agency has been more accessible, more amenable to informal calls rather than formal meetings, so a little bit more rapid response, et cetera. But in other areas has been just as steadfast and stodgy as the agency is considered to be at times. So it's hard to know. I think overall, though, with the RMAT designation, we're probably able to have greater flexibility in our discussions with the agency, more frequent discussions, which is one of the things that's promised, and this idea that if we define a phase 3 trial to which we agree, that this singular trial, if successful, will be sufficient for registration with an accelerated review.
So with all that, you're still trying to finalize some things regarding the study. Share with us what you can, what you're trying to nail down.
Well, I think the biggest discussion is around the need for the rationale behind a standard of care arm. So when Baxter started their phase 3 trial, which was done under a SPA, a special protocol assessment, they agreed at the time, remember that's 4 years ago, with the agency that they would have a control arm, which is people who receive a sham procedure but no cells. They would have obviously an active arm, and they would have a standard of care arm, which is people who received none of the procedure. At the time, it was thought that there might be an implication, a safety implication for these patients for receiving low-dose GCSF and then having an apheresis, among other things. Since then, there's been quite a bit of work that's been done and published that shows that those patients don't seem to behave any differently. And in fact, in the phase 3 program, the standard of care arm did statistically significantly worse than even the control arm. And so, I guess the FDA's first reaction was just repeat the Baxter protocol. And our response was there's a medical, ethical, and cost implication to having a standard of care arm that we think is no longer justifiable. And we're working with them to come to a compromise that will allow us to ensure that a single trial will answer their questions and get us a registration.
Great. So let's move on to the CMD study. It's a phase 2. And just maybe highlight for us what the potential difference is, in having the patients that we typically think of as having coronary arterial disease who don't have that in this population. Are they more, as it pertains to angina, does that make them more difficult to treat, less difficult to treat? How are you thinking about that?
So I would characterize CMD as a really insidious disease for a number of reasons. One is that until very recently, it wasn't easily diagnosed, and of course there was no treatment for it. It's a disease where patients present themselves with debilitating angina, typically to a cardiologist, who then goes through the normal screening process and ultimately gets to an angiogram and comes away scratching his or her head saying, the typical cause of this kind of debilitating angina is a blocked coronary artery. So they're looking for something to stent, they're looking for something to bypass. They've already determined that typical anti-anginals aren't working. And when they find clean arteries, they don't really know what to do at that particular point. So these patients then, for the longest time, in fact, because there is a a preponderance of female patients here. It's not exclusively a female disease, but probably a roughly 60-40 or 65-35 preference toward female patients. For the longest time, this was actually attributed in some circles as female hysteria or psychosomatically induced and not really a physiological disease. With the development of the measurement of coronary flow reserve, we now have a quantitative measurement that shows that these patients are debilitated and challenged when it comes to heart perfusion. And now we know how to diagnose the disease, and hopefully with a treatment that actually increases angiogenesis of the microcapillaries in the heart, a mechanism to treat it as well.
Well, great. Well, there's more I'd like to ask you, but we're out of time, so thank you so much.
Appreciate it, Kay, and thank you again for staying to the end of the day, folks.